Welcome to the Fungisphere webinar series, brought to you by the Sydney Infectious Disease Institute here at the University of Sydney. I'm Associate Professor Justin Beardsley, and it's my pleasure to introduce these talks, our passionate team, and share some of our mission with you. We're dedicated to increasing our understanding of all things fungal, be that human health, animal health, agriculture, ecology, or even the productive and industrial use of fungi. We are seeking to uncover the threats and the promises offered by these remarkable organisms. This webinar series is more than just a series of talks. It's a platform to bring experts in the field together to share their knowledge and hopefully to build new connections. It's a testament to the synergies that exist between researchers here, but also colleagues globally. Through these talks, we share some of the incredible work being done here in the University of Sydney, but also provide a platform for international experts to share their groundbreaking research too. So, whether you're a seasoned researcher, a student, or simply somebody with an interest in the fascinating world of fungi, you'll find something of interest in this webinar series. Together, we're going to explore the threats and the promises of the fungal world. In this talk, Dr. Tien Huynh from RMIT in Melbourne shares some of her research interests. These range from upcycling through bioremediation and the production of medicinal products from fungi. Let's hear from Tien.
Thank you, Justin. And yes, my great pleasure to welcome Tien here today to talk to us about her work. She's coming to us from RMIT. We just had a quick chat about Tien's background, and she actually started at RMIT doing her undergrad there, and she did a PhD at Melbourne Uni with, among other people, the great Tom May that so many of us know very well. And she was then working on orchid mycorrhizal fungi. So that was her first sort of love of fungi triggered at that point. She then did some postdoctoral fellowships in England and Italy. And then she returned to Australia and she actually started to do work on cancer, but developed an allergy to rats and ended up coming back to the fungi. So good on you, Tien, for recognizing that's the place to be. And she has a lot of passion for fungi, which I'm sure will shine through in her talk. So thank you so much for coming to give us this talk, Tien, and we're really looking forward to hearing about it.
Thanks, Dee, and welcome everyone. It's my pleasure really to share a little bit of the research that we have going on at RMIT. And like Dee mentioned, I've had quite a diverse experience in terms of both Australia and overseas and a wide range of things. And my first love started with plants and flowering things, but along the way, I also had a lot of dealings with fungi, and now I'm working a lot more on them, and I just absolutely love it. And I apologize for the name, Fungi-Tastic research, because it's a bit geeky, but I think it really explains the passion we have and the enthusiasm we have for the opportunities that are presented to us with the fungi. I'm going to share my screen. I hope you can see this. And I really want this to be something that will allow us to have the conversation for later to talk about things that we can collaborate together and maybe even start a revolution to make Australia the fungal hub. So first of all, I'd like to acknowledge country, the Wurundjeri people. And in here, I've got a picture of the orchids, the native orchids that are found in Victoria. These are called the Caledonian orchids. That one's a Caledonian.
Sorry, Tien, we don't see your screen yet, sorry.
Oh, okay, hold on. Do I need to do anything?
Yeah, just click share screen again.
Okay. Okay. Sorry, everyone. Can you see my screen now?
Got it, perfect. Thank you.
Okay. So these are the orchids that are found in Victoria. And I first started falling in love with them during my PhD. And that led on to the mycorrhizal fungi within them, because the orchids, they survive in very dry, nutrient conditioned environments. And the fungi was actually really good for them to obtain the nutrients, spread out their network underground so they can obtain those nutrients and bring it into accessible forms for the orchid. And what we really focused on was the environmental impacts, so specifically fires, flood, rain, and all that sort of environmental changes that was happening in the natural landscape. So the main topics that we had was obviously fire, climate change, and we've had quite a few publications in terms of mycorrhizal fungi. And I've got a picture there of a scanning electron microscope of the fungi that's found within the epidermal cells and the cortical cells of the stem collar, which is at the base of the leaf, where all these fungi are concentrated. And what happens is the orchid breaks them down to obtain those nutrients. And you can see the stages of the fungi where they're actually broken down to supply those nutrients. And so what we were doing with this was looking at where the fungi was the key thing that was contributing to the success of the orchids based on the sites that we were looking at. So they were remnant sites versus reintroduction and translocated sites, because a lot of our native bushland undergoes a lot of changes because of our needs and desires of things that we need to do for it. So we were noticing a lot of main roads being put in. So the sites where these orchids were located were being disturbed, and a lot of the plants were being put into areas, so translocated to areas that may not be suited for the fungus, and so the orchid doesn't survive. So that conservation effort is fruitless, really, if we don't know what's going on below the ground, and specifically for the fungus. We need to support that as well as the plant. So we looked at both rare and common orchids and the fungi that were associated with it. We're going to have a few more publications this year, so I'm ready to submit another one for climate change, looking at specifically stratification and temperature changes based on historical meteorological data to give us a more future planning projection of the temperatures that are going to be needed to encourage the growth of these orchids that we're trying to save. And along the process, I had a lot of interest in overseas projects. One of them was on environmental sustainability. And it brought me along this path of looking at mushrooms. So what you can see here is mushroom compost on the left, and on the right are the mushroom bricks that are left over after they grow them for food. So this one I think was straw mushrooms and oyster mushrooms. And this one is the lion's mane, which is believed to be good for the brain. So we've also started working with a lot of the farmers overseas growing more exotic medicinal mushrooms to see if they actually do what they say. Because with my medical background, I was able to extract the mushrooms and test them. So I do a lot of outreach activities, and that's why I put the little girl there, because she was just lovely. She really liked the fungus. She really liked the mushrooms, and she wanted to know more about it. And I love encouraging young kids to actually get into this, because I do think the mushrooms provide overseas, there's a lot more cultural acceptance. Where if you go to the farms at the local farmers market, you'd have at least 20 different species of mushrooms growing locally. Here in Australia, I've noticed that especially with the supermarkets, we only probably get button mushrooms, and sometimes you might see oyster mushrooms, but that's probably the diversity that we have. You might see a bit more in the Asian grocery markets because it comes from that cultural acceptance of fungi and mushrooms to eat in our diet. And the foods that these farmers made for us was just amazing. I had a deep fried oyster mushroom tempura type like thing. And it was the most amazing thing, because mushrooms on their own, when they were eaten fresh, they were very nutty. But when they were deep fried, they were very creamy. And it was like a dessert. And I was really quite astounded with it, because we tried different types of batters. And when they put it in, for instance, they had coconut and sesame seed. It was just really fantastic. And they're so keen on changing perspectives and doing new recipes to get the public involved in this sort of thing. And then once the mushrooms have been picked, they actually recycle a lot of the waste that's produced. And it's this closed loop cycle where it's a circular economy. And if you see in the background on the left-hand side there, all the compost gets put in and they grow crops on it. And there was very little waste. And I really liked that component of it. And they centered mushrooms around this. Back in Australia, we also did a few other projects, which is fungal enzymes, looking at the ability for these enzymes from these fungi to reduce tissue culture or hydroponic systems, so specifically Phytophthora and Fusarium. And we found that some enzymes from these fungi were actually quite good at inhibiting the growth of these pathogens. And we also looked at its ability to break down different tissues, for instance, were inoculated with Phytophthora. So that was interesting, and we haven't published that yet. So I'd really be keen to hear anyone who's interested in continuing that further to look at some genetic work to see if we can actually see the genes that are changed due to the fungal enzymes. And then we had some businesses contact us because we were starting to publish quite a lot of work, and we had businesses very keen looking at sustainable packaging. And they contacted us and gave us a challenge, said, hey, can you produce a sustainable mushroom packaging container for us? So I got my students to work on that, and they produced these square-like things, which you could put. So the truffle industry actually contacted us, and it was quite an interesting time because the meeting was set at a 5-star restaurant by a couple of hatted chefs, and they made us eat all these truffles, and we're feeling very, very privileged because it was truffle season. Some of my collaborators had never had truffles before, and that was just amazing. But we were talking about this sustainable packaging from the fungi. And the students were, so I've got the students there, Matthew and Patrick, and they decided to, so the brief was to look at input material that the fungi could grow on. And the criteria was that the input material had to be sustainable waste that we're throwing out. So they decided that they wanted to do wheat and sugarcane bagasse. So every morning they came in, they'd say, oh, we're going to the sugarcane drinking place because the sugarcane producers were squeezing out the liquid for this. So they'd come back with some sugarcane drink for me and the sugarcane bagasse. And they would take it home and get their parents or even grandmother to cut it into little pieces because they realized we couldn't put it through the mulcher because the sugarcane bagasse was just too fibrous. And once they sterilize it, they put it into just plastic containers and different container sizes, and they grew these boxes. And when they actually cut it down to the size that we can measure the strength and tensile, the tensile strength and pull, we compared it with polystyrene, which is what we normally use for packaging. And we found that it was the fungal bricks or containers that we were making was 5 times stronger. So that was certainly interesting because it did feel strong. We tried to break it by throwing against the wall, and we weren't very popular with it. But everyone was quite interested in the way these boxes are made. But if you ever look at it, it was not very attractive. So we did send it to some art students and said to them, can you do something to this to make it look much better? And we're still waiting for them to come back because this is just at the beginning of COVID. So we haven't been able to continue with this because it's just been a bit of a trial to get into labs and grow weird, funny stuff. How am I going with the time, Justin?
Good time.
Good. Okay, I've only got a few more to go, so I should take my time. I also had a PhD student who's now graduated, Mitchell, who looked at fire retardant properties of fungi. And what he did was he grew these things we call fungal bricks. And you can see them on the right-hand side here. It's just like a brick, a square thing. And we compared it to polystyrene in terms of how it can resist fire. So we put a blowtorch onto it, and at the end of, I have a time-lapse video, but apparently the media is not loading, so you can't see it in real time. But what we did was that after 30 seconds, the fungal brick still maintained its structure. It produced no gases or smoke compared to the polystyrene brick there. It was much, much better in terms of a fire retardant material. So we found that not only was it effective in terms of the temperature going through the sample, the temperature didn't change much. It was safer because there was less toxins being produced, the carbon monoxide, carbon dioxide, as well as a whole heap of other compounds that were released into the air, as well as smoke. There was no smoke that was produced. It just charred on the surface. But if you look at the back, it's still maintained pretty much intact as when we put it in at the beginning. Now, the important thing for a lot of these products is whether it's cheaper than what's already existing. And we did find that it was cheaper because the material that we were using was from waste. So this used rice hulls and sugarcane bagasse, I think it was. We also used wheat straw. So they were all very sustainable products that were easy to access, and Australia produces a lot of it. So that actually was quite good because we did a feasibility analysis in terms of, well, is this feasible? And we found that it was up to 31 times cheaper than synthetic foams and engineered woods, which is what we normally use at the moment for fire-resistant building products. So putting it in as cladding within the walls. We found that compared to the polystyrene and the particle boards that's been used, it is obviously much cheaper, that you can see there. So for the mycelium composite, the most that it would cost is $90 per metre squared, compared to the other things that can go up to $630. We found that it was lightweight, more than all the other things that we were using. So some of them were going up to 1,500 kilograms a metre squared. So it means that you would need less reinforcement to hold these things up. So it was 7 times lighter than the lowest level. And we also found that it was safe. So there was less heat, less smoke, and toxic gases that were produced. And it took much longer to catch fire. And even when it did, it just charred on the surface. And obviously less smoke produced, which is one of the main things that causes fatalities in house fires, because with the smoke, you just can't see and you get intoxicated with the fumes. I've now got a student working on this, looking at advanced composites. So looking at the automotive industry, so lining it inside the cars for sound absorbance, as well as for thermal protection in airplanes, because it's a lightweight material. So we're looking at improving travel, which is probably a moot point now, but this started before COVID. But we were really looking to implement this fire resistance, lightweight, and the strength properties of the different fungal composites into the things that we use and love. I also had another student, and she's yet to submit her thesis, so we really want to publish this at some point in time, looking at the sound absorbance. So again, we produced these bricks, which was very simple to make, and we put it through an impedance tube where you put your sample at this end. The sound source comes in from the left side, and you can measure it coming back in terms of sound that's reflected or absorbed. And what we did was we tested on several different non-toxic, so medicinal mushrooms that are edible, so Trametes, Pleurotus, and Ganoderma. And we had different types of inputs. So we had rice husk, which is thrown out, wheat straw, and sugarcane bagasse. And if you look at the one that I've got circled, so these oyster mushrooms on wheat straw can actually get up to 100%. So on the left-hand side there, the one means 100%. It can absorb 100% of that sound at that wavelength. Now, when we compared it to what's normally used for sound absorbance, so in your homes, for instance, especially for high rises, at the wavelength that causes us the most annoyance is traffic noise, which is usually below the 1,000 hertz range. So I put a square around those boxes, those areas there, and you see that at less than 800, we're getting only about 20% absorbance of the sound. So there's still quite a lot of sound that we're hearing as we're sleeping or as we're just sitting there. And over time, you don't notice it, but it does cause a lot of health issues. And especially when we become more urbanized, we're starting to grow, have houses built on top of each other, and we're becoming more condensed. It's going to be a real problem in terms of our ability to sleep, sound sleep, as well as background noise that we're not hearing. The mushroom or the fungal bricks that we have, so the Pleurotus on straw, you'll notice that less than 800 or 1,000 hertz, you can get up to 60% absorbance. At higher wavelengths, you can get up to 100%, but that's not really the sound that we're really focused on. But most of the traffic noise that we have is less than 2,000, but the background noise is less than 1,000. And we found this really, really great because what we're using is probably not the best product. And all the products that we're making with the mushroom or the fungi have so much potential, and we're using waste products that could actually help us in terms of environmental sustainability. So in summary, that was probably more than 3 times better absorbance for less than 1,000 hertz, which we found really great because we've got the data there to show so many benefits from using fungi. And then we also have projects with edible mushrooms, especially for medicinal. So I'm actually collaborating with people now in China and Taiwan, looking at the microbiome in terms of whether fungal diversity in our gut is important for health and different lifestyles. So hopefully that will be published soon, which I think might happen mid-year, hopefully. But along the process, we decided to do some fun things and grow our own edible fungi. So this is me in the lab. So we had some tobacco research going on, but I decided to throw my little mushroom things that we made up. So we had a lot of Pleurotus mushrooms that everyone took home to eat and stir fry. We had these pink oyster mushrooms as well. And we had a lot of samples from overseas that were sent over to look at the medicinal applications. So wood ear mushrooms, lion's mane, as well as some unknown ones. If anyone knows what that one is, please let me know. We didn't collect that one, but we've also got straw mushrooms that were actually very popular over there. But we want to look at the medicinal applications of them. There's a lot of big farms in Asia growing mushrooms for medicinal applications, especially for the Chinese market, specifically Ganoderma, which is linseed. And there's quite a lot of products that are being made, and we were helping them look at sustainable agriculture in terms of the input that was going in, making sure that it was from sustainable areas and practices were clean and acceptable for world standards. So we worked with a biotech company in Vietnam, especially around the areas where lots of rice were growing, and using the natural products that they have to produce different things like they had powders as well as wines, which they made me drink. And it's not to my taste, but if you like alcohol, I think it would be certainly possible. And they also looked at growing them and using different light sources to see if it can actually increase in terms of vitamin D and different levels of nutrients. So we've got some research that we've started to do looking at the bioactive compounds as well as its bioactivity. One of the things that was really interesting were the cordyceps, which is we call the zombie fungus. And there's a lot of research going on over there in terms of producing these, because it's quite an interesting story, because these in the Himalayas, if you get them from the insects, they're worth quite a lot of money, and people make their whole careers out of this, which is quite interesting. But a lot of research has been going in the background with growing them and looking at the different enzymes that allow for the cordyceps to grow and fruit from the insect bodies. So the products that they were producing, they wanted us to test in terms of bioactivity and having a quality control in terms of the cordyceps, and which is the active ingredient that comes out from them. So that's the end of my talk, but I would really like to acknowledge both the students and industry that have been involved in this. And it's been an amazing journey, because we've just been astounded with all the different projects that we've had with fungi, because they're vast and varied, but there's so much potential. And I think there's so much more we can still do. And especially with the sustainability component of it, I think we can really make an impact to not only have great foods that we can eat that are beneficial for us, but also do well for the environment. So thank you for listening.
Tien, thank you very much for your talk. Extremely interesting. Okay, I'm just going to change the view so we can all see each other. So a couple of recommendations have been added to the box for the ID question. Obviously, nobody here had any idea, but some possible people you can see in the chat box that might be able to help with IDs. And now I'll invite any questions from the audience. Is there anyone who wants to unmute themselves?
So we've got one. Sorry, Julie. I'll let you do it.
Oh, okay. Tien, thanks for a really, really fascinating talk. These fungal bricks seem to tick all the environmental boxes, so you're really onto a winner there. But I wondered if you could talk a little bit about how you actually make the bricks, and what are the biological processes that contribute to you know, the toughness of the bricks. So I know that fungi, like they secrete a lot of extracellular enzymes, and they're obviously breaking down all the different waste materials that you feed them. But why doesn't it all decompose into sort of nothing? And so what are the biochemical products, I guess, that give the mechanical strength to the bricks and the fire retardant properties as well?
Okay, so we had a whole load of fungi that we looked at, and they do produce different types of structures, so skeletal structures, and some of them can actually form these, we call fused plates. So they're no longer hyphae as you would normally get them as individual strands. Once they start to fuse, they do form like a protective coating, and it helps them with the water resistance as well. So we haven't looked in terms of the actual cellulose and cellulase activity with the different fungi and what actually makes them strong. We have done the mechanical tests to see them. And a lot of these fungi, when we're growing them, you just see that even when they're fresh, they're leather-like. And there's been a lot of companies that produce these fungi as leather alternatives. Mm-hmm. And the structure that's, if you know leather, it can be very waterproof. It's very strong in terms of if you try and break it, it just doesn't. Once you dry it, that depending on the property it has, it does actually become much more, well, very tough. I mean, I've got a brick here with me, I can show you, but it's, if I throw it on the ground, it just won't break. And I've had this brick with me for 5 years and it won't degrade as long as you take the moisture out of it. And I think someone said the chitin walls. And yeah, so once they form their structures, if you take out the moisture, it will just keep it, retain it. Does that answer your question?
Yeah, so I just wondered then why you actually need the biomaterial and why you just don't add. I guess you're just giving them fuel basically to make more fruiting bodies. I guess that's the take home message. Yeah.
We have tried to, we've grown them, so for the plane project to make the lightweight material, we've grown them on liquid culture, so they don't actually need a structural substrate to grow on. You can actually grow them on top of water and it'll grow fine, and you still get that leather-like, I mean, they're quite thick depending on how well the hyphal strands can actually connect to the carbon source. And what we've been trying to do is put them on a layering, rastering thing so that the fungi grows, and then we just evaporate out all the liquid so that you get that thick and dense structure. But if you grow them on a substrate already, it's easy to absorb the nutrients so that the fungi can grow within that and then break it down as it grows.
Thanks. Yeah, you had a question there about the residual antibacterial, antifungal activities. Would you like to just unmute and ask yourself?
Well, it just occurred to me, is there something extra you could get from the use of this packaging for food storage that might prevent spoilage of it, for example, due to bacteria in meats of one sort or another? I presume antifungal activity is not exactly realistic since we're producing these compounds from fungi, but I just wondered if you'd explored that possibility about packaging.
No, we haven't explored it because once the fungi have grown to the mold size, we pretty much just dry it and kill it. And And then with the packaging one, I don't have any left over because after we finished the project, we wanted to see how it was breaking down in the garden. So I put it on top of all my orchids and it broke down as soon as you put some water on it. So it's quite inert. Once you dry it, it's pretty much dead. It doesn't produce any enzymes or anything. And as long as you put water to it, it just breaks down and it's totally decomposable. And that's why I think as a sustainable packaging material, it comes from waste that we're throwing out, and then we can reuse it as a mulch just simply by adding water. I thought it was quite brilliant, really. I was just like, oh, we should have more of these projects. We don't have to clean up after you. You just chuck it into the garden and it just fertilizes everything. Yeah,
no, I think it's a really exciting presentation. Thank you.
Oh, thank you, Tanya.
Justin, can I ask a question?
Yes, please.
Yeah, the very nice talk. I always was wondering, I have read about this for a number of times that people use this for different products, but as you mentioned, you take the water source out and then you try it and it becomes permanent. But are you really sure that because you have fungi having spores, and even if you take the water out, how do you actually, I always was wondering, how do you prevent that this fungus will not start to sporulate again later and then grows out of whatever? Like I normally, I can understand if you take the water out of the mycelium that it may be dying and totally dry, but a lot of the spores can actually survive actually quite long in very dry conditions and maybe come up again. So how do you make sure that any of these things are not starting to grow again when they get rehydrated?
When it rains, you mean?
Very good question, Wheeland. And that's why we spent a lot of time selecting the right fungus.
Yeah.
Fungi that are the spores are very hard to break down, and they will resist a lot of, treatments. So that's why we chose medicinal fungi that we know won't grow in certain conditions.
Okay.
And spores, and even, so we chose pleurotus, so oyster mushrooms. And I haven't seen them grow back up because I was happy to have oyster mushrooms growing in the gardens, but it hasn't come back. And we know that even after drying, we've left them for years. Well, the labs is quite a moist environment, and I've left some in the growth rooms and nothing's come back once we've dried it. And that's why in our initial study, we had a whole range of different fungi, from saprophytes to pathogenic to medicinal and all that stuff. And we chose specifically the ones that are medicinal, so Ganoderma, Trichomedes, pleurotus, we had lion's mane, we had shiitake, we had a whole range of things. And the selection criteria was one that had to be beneficial. So if it does spore back, then at least, it's going to die out in the cold anyway, because Melbourne's temperature is very variable. And two, that it will grow fast so that we can finish the project on time. And three is that if it does spore, then we can use it. So that's why we chose those three. And there's no problems with students working with it, because even the spores, if we have toxic spores, we're breathing it in, it's not a sustainable thing for manufacturing, because we need to consider the workers that will work with these products. So we're not just looking at the end product, it's affordability, it's functionality, but also the initial input stage that's needed. So a lot of the fungi we chose will grow on very common waste products. So they're not fastidious, they're just, happy to grow most things as long as it's carbon source. And that they're not pathogenic, so that whoever has to handle with them, it's not going to be a problem.
Yeah, I was much concerned about being pathogenic. I was just thinking how the structure will be sustainable afterward if you build them in, like, for example, the idea is to build houses from them.
Yes.
How you make sure that the house stays in 10, 20 or so years, that's the kind of thing. It's not starting to grow again. But okay, makes sense that some of the fungi are maybe not so resistant than others, and if you use the right ones, they will be not growing anymore. Okay. Thanks.
Fantastic. There's somebody working in this area. Do you two things to say. One, as a potential person that I'll put you in touch with, somebody from University of Sydney called Jane Gavin, who works, she's from a creative background, and she works with industry in Vietnam in terms of creative ways of utilizing industrial waste. So it could be ideal, especially if you're looking for people in think about how to produce that packaging in a more saleable kind of format. And the other question I had was, have you been doing any work around bioremediation? Because that seems to be quite a kind of a growth area for this sort of research.
We did. We looked at using fungi to flocculate different mixes, but I don't think I had many good results with that because it didn't work as well as we thought, and there was a lot more effort in trying to get the final biomass out from the mix without adding things to it. That made sense. So we have been looking at bioremediation properties of fungi in water, in liquid condition, to flocculate it so you can just scoop it up and throw it out. But there's certainly a lot of people looking at that as potential, especially for biofuels even, to grow in contaminated water and then taking it out and then using it as fuel. Yeah.
Fantastic. Are there any other questions? Dee has just appeared, so maybe you have a question.
Can I ask a question? Tien, I was really interested in the cordyceps stuff because I was always under the impression that they were very valuable and expensive, and that was because they were pretty hard to grow, but it looks like they're not that hard to get growing, at least in the pictures you showed.
Well, it's a bit of an industry secret, I think, or a myth really, because if I can grow it, and I'm pretty bad with being there every day to look after them. They're like plants, they need watering every day. Because I think there was, because I spend quite a lot of time overseas teaching outreach programs. And I had one of the mushrooms growing in the growth rooms, and I came back and I had it in a glass jar in the fridge. And it had grown out of the glass jar and contaminated the whole little growth area. But cordyceps over there was they use red rice, so it's a very specific ingredient, and they were actually able to. So what they did was also get the insects, grind it up, and isolated out the enzymes that were in the insects, and made an insect mush as well. But it would grow equally as well without it, using just what's available around. As long as you have clean cultures and you know what you're looking for. And they were able to even inject the fungus into the insects, and that would grow out.
Right. So that picture that you showed with the kind of glass jar with all the cordyceps inside it, they were just being grown inside that jar, were they?
Yes. And they also had ones growing in just jars normally with rice grains. And what they did was they also, once they grew it, because people like to see it, and they gave me a whole heap of samples to trial out, they would actually mix up wine with cordyceps and then put that liquid in on top of that so it looks like it's growing from the cordyceps base. Quite fascinating, really, because it's quite a prized product, especially you can, because over there there's a conception that if you can see it growing from the insect, that's more valuable than it growing on its own and having a clean culture. But I think there's a lot of benefit for having clean cordyceps because, it hasn't gone through the insect. And it tastes the same to me.
So you tasted it?
Yeah, and they also made food from it. They made me drink cordyceps wine, cordyceps alcohol, cordyceps caves. I was cordyceps out. It's just like any other fungus, really, but with a slight carroty aftertaste.
Huh, interesting.
It's great that there's a clean alternative. Working in Vietnam, we used to see people who'd harvested cordyceps that weren't cordyceps, and they get really sick because it's actually a toxic thing that they've collected. So yeah.
But we have Australian cordyceps as well, and I'd really like to be able to do that because we can replicate that here with the waste that we have and sell it as a very sustainable product that's clean and beneficial if we can prove the bioactivity. So that's my challenge. If everyone wants to get involved with Australian cordyceps.
Sounds great.
Thank you.
Do you have any other questions for Tien? No? Do you, are you up to date with what's happening with psilocybin research in Australia as another potential major product from fungi?
I've heard a little bit about it, but I'm not really keeping my eye on it because it's something, you know, I've got so much going on at the moment with the bricks and stuff, because we're always getting, we had a collaborator contact us for fire doors before COVID, but with COVID happening, everything's only starting up now. So I think it's a really good chance to finish some of the projects that I've got there, which most haven't been published, to just get it out, because I think the opportunities are there to develop further. But certainly the psilocybin is actually good.
Yeah. Fantastic. Well, I think there are no more questions, so just remains for us to say thank you so much for the talk. It's super interesting and very inspiring. Obviously, very passionate about this topic. Thank you for joining us. I hope you've enjoyed today's talk from the Fungisphere webinar series, and we look forward to seeing you next time.